Hi again. Thank you, Dr. Kazanoff, for that informative presentation. Just again, as a reminder to everyone, you can ask questions through the Q&A button on the lower left of your screen at any time throughout the presentation. And I have the pleasure of introducing our final speaker today, Dr. Sean Eddy, who earned his doctorate from Carleton University by characterizing the role of the PI3K, AKT, and PPAR gamma pathways in stress adaptation. He subsequently completed his postdoctoral studies at Boston University School of Medicine, characterizing kinase signaling networks in breast cancer, and implicated the IKBKE as having an oncogenic role in progression of the disease. Today, he is speaking about expanded clinical research opportunities for crizotinib, identified from an analysis of over 5,000 exomes. And now I'd like to turn it over to Dr. Eddy for his presentation.
Hi, thanks for tuning in today. So today I'll talk about the potential expanded clinical research opportunities for crizotinib based on analysis of over 9,000 samples from the Oncomine Knowledgebase, and this includes over 7,000 patient exomes that have been subject to whole exome sequencing. So as part of the drug discovery and development process, there are delineated preclinical and clinical phases of research. A lot of what my colleagues have talked to you today about have focused on preclinical target discovery, target validation, and generating early biomarker hypotheses. What this talk will be focused on is when a pharma company has an approved drug, what they can do to potentially extend the label or the indication for that drug, opportunities that we can help with, rather than generate a clinical trial from scratch to try and identify new opportunities. So to do this, we really take advantage of the Oncomine Knowledgebase. This is a knowledge base of expertly curated genomic cancer data that's been collected, curated, and assessed over a number of years. And as I noted before, it includes over 90,000 samples. And this includes both gene expression data, copy number data, mutation data, gene expression signatures, fusions, as well as cell line data from over 2,000 cell line models and a number of tumor biomarker data points. So this all gets fed out into the Oncomine applications, which a number of our pharma partners subscribe to, as well as consumed in-house for collaboration work by the Compendia Bioscience Translational Bioinformatics Services team. What this team does is we really leverage our experience and expertise in cancer genomics that we've built up over a number of years, both with Thermo Fisher and employment through other previous employment, and really leveraging our ability to dig into the Oncomine Knowledgebase and pull out relevant cancer genomic information that we can then translate to our partners to help them in as they're prioritizing their portfolios, as well as doing things like indication expansion. So in this case, I'll talk about specifically about crizotinib and the potential expansion of crizotinib in a research setting. So right now, crizotinib is approved for ALK-positive non-small cell lung carcinoma, but it also inhibits activity of LTK, which is a homolog to ALK. MET, which is actually what it was originally designed to inhibit, the RON kinase MST1R, as well as ROS1. So by surveying the aberrations in ALK, LTK, MET, MST1R, and ROS1 in the Oncomine Knowledgebase, we can identify potential expanded clinical research opportunities for crizotinib, which can then expand the market for this drug and also have positive impact in treating cancer. So our analysis really focuses on digging into tumor data that we've collected and curated over the last number of years. So as noted earlier, we performed fusion analysis in over 7,000 patient exomes across a diverse number of cancer types. And here we're just noting the different cancer types that we have assessed for fusions, as well as the number of tumor samples that we have within each of these cancer types. And in doing so, we can identify known ALK fusions. So in lung adenocarcinoma, EML4 ALK has been identified in 5 to 7% of cancer samples. And so we can positively identify these fusions. We can also identify EML4 ALK occurring outside of a lung cancer. So we've noted the EML4 ALK fusions are present in colorectal cancer as well as thyroid gland carcinoma. In addition, we've identified a number of additional fusion partners for ALK in a number of other diseases. So in bladder urothelial carcinoma, we've identified fusions with TPM1 and ALK. In colorectal cancer, protein kinase A and ALK, as well as a fusion in melanoma between NCOA1 and ALK. Additionally, as we can see in figure B noted here, we find a recurrent STR and ALK fusion in both thyroid gland carcinoma as well as papillary renal cell carcinoma. This fusion has been described recently been described in thyroid gland carcinoma and as an ALK positive event, and could potentially be expanded into the papillary renal cell carcinoma setting based on this data. Additionally, we've identified fusions with the ALK homolog LTK. LTK shares a similar kinase domain to ALK and is susceptible to crizotinib inhibition in vitro. And we've identified that in thyroid gland carcinoma, we do see fusions in LTK. And these, the plots in D in the lower right indicate ALK and LTK, ALK on top and LTK on bottom, and looking at exon level expression for each of these genes. And we can see that in each case where we see a breakpoint as indicated by a dark red dot, that we see a rise in exon expression for both of these genes. This is also another indication that there's a positive fusion event going on and gives us additional confidence that these fusions are real. In addition to the ALK fusions described previously, we've also seen positive ALK fusions identified in sarcoma. A fusion with ALK and actin G2. In this case, we're seeing in 2 individual patients, and as we can see from the exon plot on the bottom, we see a very high level of ALK expression detected in one of the fused samples. So this is also another potential opportunity that could lead to potential indication expansion. In addition to ALK, there have been a number of case reports that have reported crizotinib having the ability to inhibit ROS fusion positive cancers as well. And we've been able to identify a number of these known ROS fusion partners in our fusion data, and just providing an example of a number here, primarily in lung adenocarcinoma, but also identified in glioblastoma. These fusions have all been shown to have elevated ROS activity, and consistent with what we've shown earlier, a fusion leading to increased expression past an exon breakpoint. On the fusion plot on the bottom, we have the expression data on the y-axis and exons on the x-axis, and we can see in fusion positive samples indicated in pink that elevated expression occurs post breakpoint as a positive indicator of fusion event occurring. If we take this data and then summarize it across all of our cancer types, we can see varying levels of fusion frequency occurring, ranging from 0.5% up to 2% in some cancer types, with the realization that we may be a bit more conservative in reporting true and validated fusions. In addition to fusion events, we also look at mutations to identify recurrent mutations that can lead to activation of any of the potential target genes for crizotinib. So we've integrated NGS mutations from TCGA, high impact publications, and ICGC, as well as integrating mutations from additional sources, including Cosmic and others. What we're showing here are the sample counts that we have for tumor samples from whole tumor exomes that we've collected and curated. We do have additional samples with targeted sequencing, but this is the broad whole exome, whole tumor genome sequence samples. If we look at ALK, what we can see by looking across the amino acid sequence of ALK, we identify a number of hotspots that are recurrent across a number of different cancer types. A large number of them occurring in brain and CNS cancers, which, as people may know, are primarily occurring within neuroblastoma. We also identify a number of recurrent mutations in lung adenocarcinoma as well, as evidenced here by the W247 mutation. One thing to pay particular attention to, though, is that some of these mutations are also associated with resistance to crizotinib. So if we zoom into mutations that we see within the kinase domain, a number of these mutations have been shown to lead to resistance to crizotinib. So in the case of all these mutations, when they occur within the context of a fusion protein, this leads to resistance to crizotinib. And in the case of the F1174 mutation that's recurrently identified in neuroblastoma, this mutation leads to decreased sensitivity of crizotinib relative to ALK-amplified neuroblastoma. Second generation inhibitors that are now targeting ALK have been shown to overcome many of these mutations. Looking at MET across head and neck cancers, we see two primarily recurrent mutations at tyrosine 1230 and tyrosine 1235, both occurring within the kinase domain. Interestingly, the tyrosine 1230 is enriched in metastatic samples, while the tyrosine 1235 mutation is enriched in primary samples, as indicated by the pie charts below. This may suggest some differential targeting of MET in these cancers. When we take a step back and look at all of these genes as they're piled up across cancer types, we can see that recurrent mutations in ALK, MET, the RON kinase, MST1R, and ROS1 occur up to 4.5% of major cancer types, and really giving a broad cancer, giving a broad perspective on the diversity of targets, or the diversity of cancer types that are affected by aberrations in these targets. Looking at amplification events in cancers, we see that we have over 19,000 tumor and normal samples in the Oncomine Knowledgebase. This encompasses over 3,000 samples that we can assess in breast cancer, over 2,000 in both brain and CNS cancers and in lung cancer. And the breadth of normal samples really gives us an ability to differentiate what's likely to be a cancer driver from something that may be just a copy number variant in normal tissues as well. In looking at both ALK and MET from a high-up view, looking across cancers on the y-axis and then estimated copies of these genes on the x-axis, we can see that there are a number of high copy number events occurring in brain and CNS cancers for both ALK and MET. And then also with MET, there are a number of high copy number events that occur in brain and CNS cancers. Gastric cancers is indicated in brown, and lung cancer is indicated in pale orange. And I will note that the estimated copy number that's presented here may be somewhat conservative, as tumor samples that are isolated often contain normal tissue as well. So it's not a pure tumor sample that may be analyzed. When we dig into brain and CNS cancers, if we get into the tumor subtypes on the left, we see that all of the high ALK amplification events are occurring within neuroblastoma. And if we look at the chromosome 2 landscape survey on the top right, we see there's a bit of a peak occurring in the region where we see ALK. And if we zoom that into focus only on neuroblastoma and then filter the data down to samples that have only amplification events in ALK, we can start to see that there's the tellings of a focal amplification peak, and we have this assayed over 240 samples, so we have a fairly good confidence that ALK is a focal event when we're seeing a high copy number gain in the gene. In addition, if we take a step back and look at MET, we can see that when we look at the cancer subtype level, in brain and CNS cancers, MET amplification is confined almost exclusively to glioblastoma, and certainly from a high copy gain perspective. In lung cancers, it's largely the high copy events are largely occurring in lung adenocarcinoma, but with a few events occurring in squamous cell carcinoma as well. Performing a similar type of analysis, so we'll start with the brain and CNS cancer on the left. If we look at chromosome 7 in brain and CNS cancers, we see a large peak towards the left of the plot, which indicates EGFR. However, we have boxed in red where we see MET amplification occurring. And if we zoom that out into looking at over 800 glioblastoma samples, we can see that MET is really part of a focal amplification event in glioblastoma. Similarly, if we do the same survey in lung cancer on the right, we can see the MET highlighted in the red box and then indicated in blue. And if we zoom into that region, specifically in lung adenocarcinoma, in 1,100 samples of lung adenocarcinoma, we can see that MET is the center of the focal amplification peak when we look at tumor samples that have the amplification event. Now, amplification is one way to identify potential oncogenic aberrations, but really what we want to see is the ability of an amplification to drive some type of overexpression event, which may be more indicative of an oncogenic event. So in this case, we have paired expression and copy number data in glioblastoma and lung adenocarcinoma. So glioblastoma on the left. On the y-axis, we've got relative mRNA expression, and on the x-axis, we've got estimated copies of MET. And the MET amplification events are indicated in pink, whereas events or samples with no MET amplification are indicated in blue. And as we can see, the MET amplification in glioblastoma leads to a fairly high overexpression of MET. There are also a number of samples that are indicated that have high expression but don't have amplification, suggesting other mechanisms of oncogenic activation. But it's nice to see the positive impact of amplification on expression. On the right side, we have 489 lung adenocarcinoma samples for which we have paired expression and copy number data. So on the y-axis, we have RNA-seq expression data expressed in RSEM units, and on the x-axis, we have estimated copies of MET. And again, as we can see, the MET amplified samples indicated in pink show very high expression relative to the rest of the samples, but also noting that there are samples without MET amplification that also do show some high expression, also indicating that maybe there are potential other mechanisms of oncogenic activation that could further expand the opportunity. Importantly, when we look at tumor data, we want to ensure that we're identifying things that will help our pharma partners have an impact. And amplification of MET was identified as having association with very poor outcome in both glioblastoma and lung adenocarcinoma. And while so each of these plots are a Kaplan-Meier survival curve, and we've got the survival probability indicated on the y-axis and overall survival on the x-axis. And again, MET amplified tumor samples indicated in red or pink, and samples without MET amplification indicated in blue. And as you can see, the curves that are pink or red drop off fairly steeply and fairly quickly, indicating that there's a very poor survival in this cohort. So if we look at DNA amplification. If we look at DNA amplification events across these tumor samples, we can see a varying degree of amplification. Importantly, we see high levels of MET amplification in a number of cancers, and as indicated in the previous slides, we see associations with very poor outcome. Sort of indicating that targeting this kinase successfully could have a positive impact. It's also worth noting that we do see a number of ROS amplification events, which we haven't gone into here, occurring in sarcoma as well. When we take these aberrations and then look at them sort of collectively, what we can see is that we have a range of frequencies anywhere from 1 to 6% across a number of major cancer types, indicating that the potential opportunity for inhibitors like Crizotinib is actually quite large. For example, we see in brain and CNS cancers, we see nearly 5% of aberrations in nearly 5% of tumor samples with aberrations in ALK, and another 2 to 3% with aberrations in MET. In in lung cancer, we see a number of aberrations in ALK, MET, as well as ROS1, and similarly in melanoma, we see nearly 5% of our tumor samples with aberrations in one of these kinases, as well as sarcoma. So there's really the potential for a high impact in the cancer field. There's a potential for high impact in the cancer field targeting aberrations in these kinases. And I think it's important to also point out that these aberrations do tend to be mutually exclusive with one another. So in this case, we're showing lung adenocarcinoma tumor samples, and we're showing them with a major with the major oncogenic events in lung adenocarcinoma, KRAS and EGFR mutation and amplification. The MET amplification events are independent of either amplification or mutation in KRAS. ROS1 fusions were independent of everything, and then the ALK fusions were also independent of these events. Interestingly, we do see some evidence of gain-of-function mutations in tumor samples with ALK in lung adenocarcinoma in the absence of fusion events, suggesting there may be additional targeting in addition to just targeting ALK-positive fusions. So the implications of doing this kind of genomic aberration survey is that we can really start to dig into tumor subtypes with really high aberration frequencies and help prioritize them for pharma as they're potentially looking at indication expansion. At the time of our initial analysis, we identified fairly high frequencies in cancer types with high unmet need, which including melanoma, where we identified ALK fusions and recurrent mutations in MET, pancreatic carcinoma, which had high level of MET amplification, although this was in a small sample set, and we do wish to confirm this in a larger sample set. The outcome in that disease is pretty poor, as well as hepatocellular carcinoma, which had amplification and recurrent mutations in MET, and again, these results we'd wish to confirm in a larger sample set. Since the initial analysis that we performed on this, a number of new clinical trials have opened up and actually begun enrolling patients in these indications, sort of suggesting that giving validation to these types of genomic surveys. I would like to point out that we also do see aberrations in small cell lung carcinoma, which remains an area of high unmet need, that we see relatively frequent aberrations in some of these crizotinib target genes. And so just to sum up, these results really leverage our genomic profiling data that we've collected and curated in the Oncomine platform to help provide a broadened scope of potential therapeutic opportunity for inhibitors like crizotinib. And with the availability of more NGS and big data and analyses, we can complete similar surveys for hypothesis-driven development of targeted therapies and help prioritize and expand opportunities for future therapies as well. And I'd like to thank everyone for tuning in today, and I'll take questions at this time. So the first question is whether or not this approach would be easily extendable to other drugs or drug targets. And the short answer to that is yes. We've collected and curated tumor sample data over a number of years and have a large breadth of whole exome and whole tumor genome sequencing data available to us. So we have a really broad sample set from which to draw conclusions from and help prioritize other drug targets as well. Okay, another question is, how are we identifying relevant variants and fusions? We've identified, we undertook a survey of various fusion callers and prioritized the ones that we find to work best, and then generating heuristic algorithms to predict fusion breakpoints, and then allow those and then align those breakpoints to different positions in the genome, and really, particularly when we're fusing to different points of the genome, and then so the algorithms identify the different breakpoints, and then the breakpoints align to different positions in the genome, and then we annotate those different points in the genome with particular genes, and then use that to help hone in on what a positive fusion event would be. And then secondly, going back and looking at expression imbalance data to really pull out what we think are the likely fusion events from things that might just be a noise. And then the last question we see, sort of, the analysis. The question is that, this analysis speaks to the need to multiplex variants under a single assay for crizotinib or other drug targets. Are there ways that this could be done? We've developed an Oncomine Cancer Research Panel, which is an RUO product, which assays all of these variants. While it's only a research use only product, and we see it for that, we do have the panel and the ability to assay and multiplex variants. And I believe that's it, unless I see any other questions. Thank you for tuning in today, and this is Sean Eddy signing off.
Thank you to Dr. Eddy and all of the other speakers today, Dr. Kazanoff and Dr. Urich and Dr. Bowden, for taking the time to present to us and complete this session of the Advanced Genomics in Clinical and Translational Research webinar series. I just want to take the time to also thank everyone who participated today, and to let you know that we've recorded these sessions and they should be available on the Bioconference Lab website in not too long. If you would like to learn more about the products and services offered by Thermo Fisher Scientific, you can surely visit that Life Technologies branded booth, virtual booth that we have, someone standing by to chat with you, or you can contact us directly at compendiasales@lifetech.com. We also want to take the time to invite you to join us in October for a virtual webinar, Cancer Research, Discovery, and Therapeutics, hosted again by BioConference Live. We will have a booth and a few talks there. So thank you all again today for joining us, and have a wonderful day.